Hello students. So today we are going to learn a new chapter, Hereditary and Evolution from chapter 1, part 1. In this particular part, we are going to learn the content, that is hereditary and hereditary changes, transcription, translation and translocation, evolution, evidences of evolution. Now students, firstly from the name of the topic, heredity and evolution. What is heredity? That is the characteristic which passes from one generation to other generation, that is from our grandparents to parents, from parents to us, or in sometimes certain characteristic may pass even from grandparents to the grandchildren. Now what is evolution? That is, as of certain time period the new species are formed, which may be because of various reasons like variation, formation of new species, which leads to evolution over a long period of time. So if you see the animation in the present slide, you can say something interesting. Now here you can see a man climbing a ladder, but it is a DNA ladder, which indicates that how the changes of DNA has taken place over a period of time, right? And then towards the right, you can see the second animation where the DNA strand can see which is broken or separated and is joined by a new strand. Now, because of this new strand, we can see how the new species are formed slowly and steadily over a long period of time as we come across a term called mutation, right? Or in present day, we can even have artificially engineered DNA where you can put a dancing gene, singing gene, or whichever gene of interest, or we can remove a defective gene as well. And in the second uh, part here at the lower end, you can see the DNA strand where you can see the different nucleotides ATGC, which forms the DNA strand, or in case of RNA, AUGC. Now moving further, you can see few more images are added before going to the further part. Now students, in this particular video, as always you can see in all my videos various images, animations with related to the topic and which creates a lot of interest in your topic, right? So here you can see the chromosome, lower part you can see the DNA strand. Again towards the right, the upper one, you can see the entire human structure. Then how the evolution has progressed, which you can see in the fourth slide, right from the apes to the human, right? Now coming to the introduction part of the topic, that is heredity and hereditary changes. Heredity is the transfer of biological characters from one generation to other via genes. Gregor John Mendel is pioneer of the modern genetics. So in simple words, as I mentioned in the beginning, heredity is what? The characteristic or biological character which transfer from one generation to the other, that is from parents to the offspring, right? Or from grandparents to the parents, then to the offspring. So it keeps on passing on from one generation to the other generation through genes. Now, in 1901, the reasons behind the sudden changes were understood due to the mutational theory of Hugo de Vries. Now, students, here you come across various scientists. That's why I highlighted the term which comes as a MCQ one word fill in the blanks questions. So here, the mutational theory was put forward by Hugo de Vries in the year 1901. Meanwhile, in 1902, another scientist named Walter Sutton observed the paired chromosome in the cells of grasshopper. Until then, it was not known to anyone. So initially, it was Walter Sutton who observed the chromosome in grasshopper. Further, research started in the direction of finding the nature of genetic material when it was proved that genes are carried via chromosomes. So further research, as they came to know about the chromosome, then further detailed research also indicated that these genes are carried by chromosome. In 1944, the three scientists, trio of scientists, Oswald Avery, Melvin McCarthy, and Colin McLeod, proved that except virus, all the living organism has DNA as the genetic material. So DNA is a genetic material which carries the characteristics from one generation to other generation. Then in case of virus, what will be the genetic material? It is RNA. In 1961, French geneticists Francois Jacob and Jacques Monod proposed a model for process of protein synthesis with the help of DNA in bacterial cells. So further, again two other scientists as named Francois Jacob and Jacques Monod, they had also proposed of protein synthesis with the help of DNA in the bacterial cells. Then it helped to uncover the genetic codes in the DNA. The technique of recombinant DNA technology emerged, which has vast scope in the field of genetic engineering. Now this recombinant DNA technology, in simple words students, recombinant, that is how we can uh, join up the different characteristics or add new traits to the DNA or change the DNA structure, as you saw in the first animation slide how the DNA can be reconstructed, right? That is by using the DNA technology, so which helps in the genetic engineering process. Now all of you know engineering, we got various engineers, but in case of genetic engineering it deals with the DNA. The science of heredity is useful for diagnosis, treatment, and prevention of hereditary disorders, production of hybrid varieties of animal and plants, and in industrial process in which microbes are used. So this knowledge of heredity also helps in various process as we read. So one of the important thing is in identifying the disorder. If the newborn child has any particular disorder, or even in the fetus, if the child has any disorder, the knowledge of this trait or genetic trait or heredity, that is genes, can help in diagnosis as well as the treatment, so that such disorders can be prevented. Moving further, now little bit information about the scientists. Now we came across various names of the scientists, so you should also know which are the scientists. That's why I added the images of the scientists for your knowledge. So father of genetics, who is that? Gregor Johann Mendel. Then Walter Sutton. Here the modern theory of evolution, which was put forward by Hugo de Vries, who was a botanist as well as genetist. Then Oswald Avery, Maclean McCarthy, Colin McLeod, who again carried out various experiments as we just read earlier. Then Francois Jacob and Jacques Monod. So all the scientists we just came across the images of these scientists. Then coming to the further part, transcription, translation and translocation. Now to understand this concept, first you can see few of the images which are added. So as I mentioned, you can see few images in all the videos animation which helps you to understand better. So here you can see the image down, DNA, which is form RNA or the mRNA, and from which the proteins are formed. So what that is nothing but we can see the central dogma of molecular biology which you are going to learn over here. So what is the central dogma? The DNA, which is transcript or by transcription forming RNA. From RNA, it is translated by or translation process, protein is formed. That's why you are coming across the term transcription, translation and translocation. Also you can see here reverse transcription, where RNA again the formation of DNA taking place. Clear? And one simple animation showing DNA forming RNA, RNA forming the protein. One more thing, if you note, the DNA which is formed by transcription and RNA to protein by translation. Clear? Now coming to the points. Now students, all the part of the chapter I put into points so it becomes easier for you to learn. So with the help of RNA, the genes present in the form of DNA participate in the functioning of cell and thereby control the structure and functioning of the body. So the genes which are present in the form of DNA, they are the ones which helps in carrying out all the function of the cell and also designing and controlling the structure and functioning of the body. Further, information about protein synthesis is stored in DNA, and synthesis of appropriate proteins as per the requirement is necessary for the body. Students, in simple words, what is proteins? They are the building blocks of the body. So we need proteins, right? Now where and how these proteins are made? That is by the DNA. So the term synthesis means what? To make, in simple words, right? So this protein synthesis or protein are made with the help of DNA. That's what is the central dogma, where you saw DNA first forming RNA, then the protein, and by the processes as you are going to learn further, transcription, translation and translocation. Further, these proteins are synthesized by DNA through RNA. This is called what? Central dogma. So students, now this term central dogma concept is clear to you. That is the formation of DNA to RNA, and from RNA forming the protein or the synthesis which takes place is nothing but the central dogma. Now, moving further, mRNA is produced as per sequence of nucleotides on the DNA. So, as we saw the central dogma, DNA forming RNA, that is the mRNA. Only one of the two strands of DNA is used in this process. That is why, if you students, if you see the DNA which is double stranded forms single stranded, that is RNA, where only one of the strands are used. The sequence of nucleotides in mRNA being produced is always complementary to the DNA strand used for synthesis. Now, what is complementary? Here, the term, when the DNA strand is used for the synthesis, the complementary strand is made with the help of the nucleotide which is present only in the already present in the pool. Besides this, there is uracil. As I told, it is ATGC pattern in case of DNA. That is the nucleotides adenine, thymine, cytosine and guanine. But in case of RNA, instead of ATGC, it becomes AUGC. Now what is this U? Uracil. So in RNA, it is AUGC pattern, that is adenine, uracil, then cytosine and guanine. Clear? So this process of RNA synthesis is called what? Transcription. So I added the image from the textbook 1.1 transcription. You can see how the DNA strand, which is first separated to form the RNA. So RNA polymerase which is used. Now students, always remember the enzymes which are used always ends with ASC. So here RNA polymerase is used, the enzyme. So here you can see the new mRNA strand which are joined over here. The nucleotides which are present in the pool joins up forming the strand. That's why as we read, only one of the two strands are used in the process of synthesis, right? Then here I added a simple animation for your understanding. You can see AT, UA, GC, how the ATGC is formed. So as you see here in the image, two strands get separated, right? As you can see here, and one of the strand is used. So you can see the ATGC pattern forming. And students, see carefully. A always joins with T or U, which is double bond. If you see carefully, you can see two bond, right? But in case of GC or CG, it is triple bond. Okay. Now, moving further, mRNA formed in nucleus comes in cytoplasm. It brings in the coded message from DNA. Now, students, now this central dogma, as you saw earlier, the DNA which forms the RNA, then RNA forming the protein, right? But where is this DNA present? In the nucleus. So in the nucleus, the DNA forms the mRNA. Okay, as mentioned, the mRNA formed in the nucleus. Now this mRNA which is formed in the nucleus comes out from the nucleus into the cytoplasm. Now how it comes? As you have learnt about the plant cell, animal cell, about the nucleus, right? So the nucleus has got nuclear pore. It is not completely sealed or joined. There's small opening present, which is a nuclear pore between the nuclear membrane. So through the pores, this mRNA comes into the cytoplasm with the coded message from DNA. The message contains the code for amino acids, that is which acids are required for formation. So code for each amino acid consists of three nucleotides. So it is a code where three nucleotides are present. That is why this code is called as what? Triplet codon. Triplet means three, right? Now, further, Dr. Hargovind Khurana, a scientist of Indian origin, has made an important contribution in the discovery of triplet codons from 20 amino acids. Now, students, there are totally 20 amino acids present, which are in the truest form. Otherwise, there are in all 64 amino acids with various combinations. So we always come across 64 amino acids, but in general, there are 20 amino acids. Now, for this work, he has been awarded with the Nobel Prize in 1968. That is, Hargobind Khurana got a Nobel Prize in 1968, along with two other colleagues or other scientists whom he was working with. So here you can see added small details of Hargobind Khurana about his work, what he did, the achievement, etc. If you see here, won the 1968 Nobel Prize in Physiology. He had shared with two other scientists. Who are they? Robert Hawley and Marshall Nirenberg. So you can remember the other two names as well. Here I added a simple image for understanding this triplet codon. If you see here that codons, three amino acid codons, how they are forming up. So you can see all the places they are formed in triplet 1 2 3 1 2 3, right? And you can see the tRNA, which is the transfer RNA, the lengthwise proteins which are formed. Now, moving further, each mRNA is made up of thousands of triplet codes. Now, if you saw in the image, just three of them, but it keeps on continuously forming the triplet codons. So there are thousands of triplet codons. As per the message on mRNA, amino acids are supplied by the tRNA. For this purpose, tRNA has an anticodon having complementary sequence to the codon on mRNA. This is called as translation. Now students, again coming to the first slide, that is transcription, then translation. So here you can see the single strand having the mRNA. Similarly, there is a opposite strand which forms, that is the anticodons which are formed, forming the complementary sequence. The amino acids brought in by tRNA, as we again saw the previous slide, the tRNA, here you can see the animation slide. So for your better understanding, are bonded together by peptide bonds with the help of rRNA. Now what is rRNA? Ribosomal RNA. Students, again you might have learned there are three types of RNA: tRNA, rRNA and mRNA. mRNA, the messenger RNA here which you see, which is formed by from the DNA to the mRNA. The tRNA and rRNA also are present, which helps in different ways. Like tRNA, which is the transfer RNA, are bonded together by the peptide bonds, and this is done with the help of rRNA, that is a ribosomal RNA. During this process, the ribosome keeps on moving from one end of the mRNA to the other end by the distance of one triplet codon. Now, this is called as what? Translocation. So we can see from transcription to translation, and now the third part, that is the translocation. Such many chains come together to form the complex protein. Again, as you saw in the previous slide, the image, how the amino acids are formed, joined, and then they form the protein. Now if I say simple, in simple words, amino acids are what? The basic unit of protein. So all this chain which is formed from single amino acid, they together join to form a protein. These proteins control various functions in the body of living organism and their appearance too. So it decides the appearance and the various function of the living organism. So again, from the textbook 1.2 translation and translocation, I added the image. You can see a single strand of mRNA. Now with relation to this, you can see the animation image of mRNA, where you can see the tRNA coming and joining up. It's the AUG, which is nothing but the amino acids. And first it is AUG, then you can see CUA, then again GGC. So this keeps on continuing. Just for your additional information, what is the first one? AUG. So always the starting codon of first codon is always this AUG. So this is just for your additional information. Then if you see carefully on the top, you can see these amino acids which are formed 1, 2, 3, 4, 5, 6, which keeps on continuing. And this amino acid you can see like a bead which forms a necklace, right? So you can see these beads form the protein necklace formation with the thousands of triplet codons. Okay. Now, further, living organisms can produce new individuals like themselves due to genes only. And some of those genes are transmitted to the next generation without any changes. So you can see how the new organisms are formed, or from parents how the offsprings are formed because of the genes. So you can see we may have some similarity with our parents, either mother or father, right? The daughter and son have some similarities. Now these similarities are because of what? The genes which are transmitted from one generation to the other generation. Due to this, some of the characters of parents are transmitted to their offspring, right? However, sometimes sudden changes occurs in these genes. Sometimes any of the nucleotide of gene changes its position, that causes a minor change which is nothing but mutation. Now, students, sometimes that there may be small changes. Their reasons can be many. It can be radiation, it can be chemical, it can be environmental. There can be many reasons because of which there may be mutation. What is mutation? Sudden change, right? So sudden changes takes place because of which the genes which are actually similar from parents might change, and it can be useful and it can be harmful also. So some mutation may be minor, very small, which does not affect, but some may be considerable, which can be effective and harmful. So example, mutation may cause the genetic disorder like sickle cell anemia. Okay, what is that? I added a slide related to sickle cell anemia so that you can get the exact meaning of that. Now again, I added the image from the text 1.3 mutation. So you can see the main sequence or main pattern here. By replacement due to mutation, as I told, various reasons are there. That is radiation, chemicals, some environmental changes which can lead to changes or mutation. If you see again with relation to mutation, I added a beautiful image over here. You can see the DNA strand, very complex strand, right? Here, due to sun's radiation or some radiation, that genes may get damaged. This damaged gene may be replaced by some other gene which is called a substitution mutation, and this can be useful or harmful. If it is useful, students, we can see some unique characteristic or different species being formed. That's how, as we learned in the beginning, evolution takes place slowly and steadily by the changes, or you can say gradual slow changes. Clear? Now further, you can see the image of sickle cell anemia. So you should know what is exactly it is. Now see the right side image first. A person who is having a sickle cell anemia or the disease. Here usually our RBCs, they are what? Spherical, circular, right? As you can see, healthy red blood cells. Now if they are affected by mutation, you can see what is the shape? C-shaped one, which may get affected in the individual, causing blockage. So you can see here the image of blood vessel. The normal red blood cell, they easily move through the blood vessels. There's arteries, veins, capillaries. But if you see a sickle cell which is C-shaped, which may get stuck up and can cause clotting or clogging of the blood vessel, which can cause disease related to the heart as well. So you can see here the normal blood vessels, due to mutation or some effects, it cause sickle cell, and that which may get trapped or clotted into the blood vessel. Okay, as we know the blood supplies to the heart, so in that case it can even lead to cardiac arrest, that is heart attack, if there is clotting taking place in the blood vessel. Now, coming to the next part, that is evolution. Okay, from the earlier part, that is heredity and hereditary changes. Now we are coming to the next part, that is evolution, and also the transcription, translation, and translocation. So the third part which you are learning here is evolution. Coming to the introduction part of it, evolution is gradual change occurring in the living organism over a long duration. Now, what is gradual change? Slow change. See, nothing can be formed quickly. Even if you see formation of a building, a tower takes place in a certain period of time, maybe 1 year, 2 year, 5 years. Correct? Then slowly it happens. Same way, evolution is also a process which takes place slowly and steadily. Okay. If things are made faster, what will happen? The building will collapse. Correct? This is a very slow going process through which development of organism is achieved. All these stages in changes occurred in various components ranging from stars and planets in the space to the biosphere present on the earth should be included in the study of evolution. So anything and everything which takes place in the biosphere. Now, what is biosphere? Very simple way, always learn the terms by breaking up or splitting. Biosphere, sphere, we can say the earth, the environment. Bio, life, the life present in the earth, right? So the biosphere present in the earth, which anything and everything present, right from the living organism, it can be plant, animal, microbes, or including the stars, planets, can come under the study of evolution. So formation of new species due to changes in specific character of several generations of living organism as a response to natural selection is called evolution. Now what is this natural selection? Means nature selects the species according to its survival capacity. The one which can overcome the environment, climate changes will survive. One which cannot. So after a certain period of time, which becomes extinct. Okay, so that is why 3.5 billion years ago the life had started. How many billion years ago? 3.5. Non-existent on the earth. So you can see in so many years, you can see so many changes, right from species being extinct, some are on the verge of endangered, or some on the verge to be extinct, or the species which are very few in number, as I use the term endangered, and some new species are also being seen with uh, changes present in them. If I give you a simple example, dinosaurs. Now there are no dinosaurs which are present millions of years ago, right? You may see in the movies here and there, in the cartoons, right? But those dinosaurs are not present now. They have become extinct. Or if I add few more examples like dodo, right? Smilax, which was a huge tiger species present in the earlier. So there are quite a few species. Even if you observe in this link, I had added two species discovery in the lab. So two species are discovered in the lab, which is present in this link itself. One is a bird species, Gansus humensis, which is almost 110 million years old, with the bird with the teeth in its beak. So you can see in this particular link itself. Okay. Further, at the beginning, there may have been one simple element in the ocean on the earth, and simple type of organic and inorganic compounds may have been formed from those. So it began with the uh, formation of simple elements in the ocean, on the earth, and then continuously changes from organic inorganic compounds. These species might have been evolved. So complex compound like protein, nucleic acids may have formed over a long period of time from those simple compounds. So as we say simple compounds, it may have hydrogen, carbon compound. So slowly and steadily they join to form a simple element, simple to compound or complex elements like proteins and nucleic acid. First primitive type example or type of cells may have been formed from the mixture of different type of organic and inorganic compounds. Number of those cells may have increased at the cost of surrounding elements. So these formation of cells may have taken from the beginning right from the organic and inorganic components, and slowly and steadily formation of new cells or new components from the chemicals which are present in the surrounding. Okay, moving further, again I added some images. See here the tree of life. You go right from the bottom of the root of the tree, right from a unicellular prokaryotic species, then the unicellular eukaryotic, then multicellular species. So from the bottom you can see the top various mammals, pisces, apes, amphibia, reptiles, and the evolution of human being from a apes, right? So you can see many species. Now I added one more image, simple image, where you can see the protist unicellular, from which the tree which is branching out to form various other species. Now in the chapter in science 2 itself, animal kingdom, you might have learned from porifera, then nidaria or coelenterata, ascalminthus, platyhelminthes, annelida, arthropoda, mollusca, echinodermata, and hemichordata, and then for further pisces, amphibia, reptilia, apes, and mammals. So you can see this basic chart shows all those species of vertebrates and invertebrates, right from porifera or the starfish which comes under echinodermata, the pisces, the reptiles or the amphibia, and finally the evolution of human beings, right? Now moving further, there may have been some differences among those cells, and according to the principle of natural selection, some may have shown good growth, some may have perished, which could not adjust with the surrounding. That is what, as we learned earlier, the species which can adjust to the nature, climate, conditions, they survive with the much better features, but the one which cannot adjust, they perish or they become extinct. At present, crores of species of plants and animals with huge diversity regarding shape and complexity are present on the earth, as all of you will be knowing about different species, or as I showed in the previous slide, how from a unicellular prokaryote, then prokaryote and eukaryote, further multicellular species are being formed. So there are thousands and millions of species. Animal diversity ranges from unicellular amoeba and paramecium to a man and giant whale, as you saw in the image. The plant diversity consists of various species ranging from unicellular chlorella to huge banyan tree. Now you can see chlorella is a algae species and a huge banyan tree. So you can see the difference in the range of species which are formed. The life exists on earth everywhere from equator to both poles. Organisms are present at all places: air, water, land, rock, etc. So you can see different species present in different climatic conditions, whether it is air, whether it is water, whether it is land, soil, rock, anywhere and everywhere, right? Humans have shown curiosity about origin of life and reason for such a great diversity in life present on the earth since ancient period. That is, from older times, we always have a curiosity of different species, how they are formed, or from where they begin. So different theories about origin and evolution of life have been proposed till today, of which theory of gradual development of living organism is accepted. So this one theory is most accepted in comparison with the other theory. Which one is this? Gradual development of living organism. Means what? Slow and steady development of living organism over a long period of time. Again, I added a theory which was put forward, which is called as Big Bang theory. 14 billion ago, there was a big blast in the space, and from which 4.5 billion ago the Earth was formed, and then the first prokaryotic or unicellular species, then the eukaryotic species, then the protozoa, followed by the plant species, the pisces, amphibia, reptiles, birds, humans, and other primates were evolved. So a chart which explains everything right from the billion of years ago, how this earth formed, followed by this formation of species, unicellular, prokaryote, eukaryote, and then the present day humans and other species which are present. Now, moving forward, theory of evolution. If you see here, again I added one more image, right? The unicellular species, slowly, steadily, how the species are being formed. That is the unicellular, then prokaryotes, eukaryotes, then the multicellular species, pisces, amphibia, reptilia, the primates, and finally the form evolution of human being, right? Now, first point, according to this theory, first living material, that is protoplasm, has been formed in ocean. In due course of time, unicellular organism was formed, that is the organism with a single cell, right? Gradually, changes occurred in the unicellular organism from which larger and more complex organisms were formed. So, from unicellular, the multicellular and complex organism were formed. All those changes were slow and gradual, means over a long period of time. Duration of all these changes is at most 300 crore years. It's not few years, it was long. So it is almost 300 crore years. Changes and development in living organism has been all around and multidimensional, and this led to evolution of different types of organism. Hence, this overall process is called as evolution, which is organizational. Progressive development of plants and animals from the ancestors having different structure and function, organization, is called evolution. So, as we are learning now from the beginning of the slide, overall process which has taken place slowly and steadily, right from the formation of unicellular, prokaryotic, eukaryotic, then formation of multicellular organism, whether it's plants, animals, and also now all these species are having their ancestors, right? Now, evidences of evolution. So to confirm the evolution or to provide evidences, there are few things which you are going to learn. That is, firstly, morphological evidence. Now students, what is morphology? See the image which is given in the textbook. Morphology means what? Study of body's external feature, that is outer appearance, how the animal, plants looks. If you see here, all these species external look, right? The presence of two ears, the eyes, nose, mouth, right? The facial structure almost triangular. In case of plants, the leaves, formation of fruits, flowers, etc. So observe the similarities between the given animal images and the plant. So these external features are nothing but morphology. Okay. Now I think once you understand the meaning, it becomes easier to learn the concept. Various similarities like structure of mouth, position of the eyes, structure of nostril, and thickly distributed hairs on the body are seen in animals, whereas similarity in characteristic like leaf or shape, leaf venation means the margins and the veins we can see. Uh, Leaf petiole, that is the stalk part, occurs in case of the plants. This indicates that there are some similarities in those group, and hence it proves that the origin must be the same and must have common ancestor. Because of this external characteristic, as we learned, the position of the eyes, the mouth, structure of nostril, distribution of hair on the body, in case of plants, the venation, that is the leaf vein shape, all these things gives an indication this all these have a common ancestor, or they originated from one single ancestor and then it has been diversified further. Second is what? Anatomical evidence. Now students, first was what? Morphology, external. Now anatomical. What is anatomical? From the image it is easier to understand. The bones, the bones of the hand in case of human or ox, or in case of bat, in case of whale. If you see the anatomy part is almost similar, only the shape of the bones might differ. So you can see the image of an ox over here, that is, if you see the forelimb, the human hand, that is forelimb of the human, the forelimb of the bat which is stretched out, and then the whale which I highlighted over here, right? Coming to the point, if you carefully observe the pictures, there doesn't seem any superficial similarity between human hand, forelimb of ox, flipper of whale, and patagium of the bat. What is this patagium? Students, if you see the wings part, the extended wing part present is nothing but the patagium. So if you see the images externally, they are different, but the internal anatomy part is almost similar. So similarly, use of each of the structure is different in respective animals. That is, in case of ox, for moving around, walking; hand for doing various function; for bat, flying; for whale, swimming in water, right? However, there is similarity in structure of bones and joints in the organs of each of these. This similarity indicates that those animals may have common ancestors. Now, third one, vestigial organ. Now, what is vestigial? The organs which are present in our body, but they are of no use, or it may be useful in our ancestors, right? So, coming to the points, degenerated or underdeveloped useless organ of an organism are called as vestigial organ, which are not useful presently. So, in living organism, sudden development of new tissues or organs for living in changing environment is not possible. Instead, existing organs undergo gradual changes. So whatever organs are present earlier, they might not be useful now, and the organs which are present now, which is according to the need of the present uh, climatic changes, present environment, present surrounding. So mostly a specific structure in the body is useful under certain situation. However, same structure under different situation may become useless or even harmful. So some others may be useless, or it can even be harmful, which we'll be learning further in the next slide. Which are the useful ones, harmful ones, or which is not useful. Such structure begins to degenerate under such situation as per the principle of natural selection. That is, one which is useful will be continued, one which is not useful will remain as it is or become useless, or it may be not present or may be changed. In thousands, it takes thousands of years for a structure to disappear. So for a change to disappear, it takes a long time, that is almost 1,000 years. Such organs are seen in different phases of disappearance in different animals. Such organ, through non-functional, though non-functional in certain organism, it may function in other organism. That is, it is not vestigial in organs. So certain organs may be not functional, they may be present, but certain organs may be present, but they may be not functioning or useless, like appendix, which is useless to human, is useful and fully functional in ruminants. Now what is this ruminants? That is the cattles, right? Cows, buffalo, ox, all these are what? Ruminants. So it is useful for them in the present age as well, but not useful for the human, and it can be harmful for human as well in certain cases if the secretion of the appendix takes place. So that's why we come across the operation of the appendix, or the appendicitis is caused by because of the secretion by the appendix. Similarly, muscles of the ear pinna, which are useless to human, are useful in monkeys for movement of the ear pinna. Clear? Now, the last point of this, various vestigial organs like tailbone, that is coccyx, wisdom teeth, and body hairs are present in the body of human being. So that's why I can see the images, vestigial structures, human external ear muscle, still it is present, but they are of no use, but is used in the apes and monkeys. Then the wisdom teeth, you can see I have added a labeled diagram, wisdom teeth, which is present at the later end of the mouth region, molar teeth, the incisors, canine, premolar. Then here the appendix, which is present inside the body at the end of the small intestine and beginning of the large intestine. So you should know where is this position of the appendix. Here it looks larger, but the position is wherever the small intestine ends, at this position as I'm highlighting, and the large intestine starts. At the junction of this, you can see the appendix. Okay, and this has to be removed if it even secretes in certain persons. So then it may cause appendicitis, that is inflammation of this appendix. Then the tailbone, if you come touch the back lower end, you can feel a small bone extension, which is the tailbone, or which is called as coccyx or coccygeal bone. Okay. Then fourth one is what? Paleontological evidence. Now, what is paleontology? If in simple words, like fossils, whatever is remaining remains of the earlier, which may get buried in the soil. Coming to the first point, large number of organisms get buried due to disasters like flood, earthquake, volcano, etc. Remnants and impression of such organisms remain preserved underground. These are called what? Fossils. Study of fossils is an important aspect of evolution. So if you want to know about the evolution, these fossils helps us very much to know about which species were present in the earlier times, or whatever things were present in the earlier times. Now, carbon consumption of animals and plants stops after death, and since then only the decaying process of C14, what is C? Carbon 14 occurs continuously. In case of dead bodies of plants and animals, instead of remaining constant, the ratio between the C14 and the C12 changes continuously, as C12 is a non-radioactive part. So both C14, C12 are carbon, but the different carbon component, or in simple words, you learn in chemistry part of it, like isotopes, right? The time passed since the death of a plant or animal can be calculated when measuring the radioactivity of the C14 and ratio of C14 to C12. So difference in this ratio present in their body. This is called what? Carbon dating method. So that difference helps us to know how many years old is this particular species, plant or animal. Now it is used in paleontology and anthropology for determining the age of human fossils and manuscript. Once the age of fossils has been determined, but by such technique, it becomes easy to deduce the information about other erstwhile organism. Means once you know about one particular fossil, we can easily relate and learn about the other fossil which are other species which are present during that period of time. So it seems that vertebrates have been slowly originated from invertebrates, which just now, as I mentioned in your chapter Animal Kingdom, we are learned across the presence of invertebrates right from porifera till hemichordata, and then how the formation of the vertebrates begins from pisces to mammals, right? Now I added all the images over here, like paleontology, study of fossil record, which is the meaning of paleontology. Paleont, the fossil, logy, to study. Then the fossil record, which you can see over here, there is all the fossils discovered on the earth. uh, the numbers, there are many here, some of them are shown. The fossil record can help scientists figure out what species no longer exist look like when the organism were evolved. So with the help of these fossils, we can make note of the structure, shape of what animals were present at that time. So here another image which is added, which is given in the textbook as well. Then few more addition of fossils remain imprints or traces of prehistoric organism that can tell when and where organism once lived and how they lived. Okay. Now again few information which is given in the text, that is 1.8. structure of ground level and fossils. So in ground level, Cenozoic era, you may find fossils of aves, that is birds and mammals. Reptiles, Mesozoic era. Reptiles, amphibia, pisces, and also the invertebrates. All these three layers shows the Paleozoic era. So here, as we just learned in the previous slide about carbon 14, which was studied by Willard Frank Libby. So it is Libby's luck or the good fortune in many stages of this research was mostly miraculous. So that is why it's mentioned Libby's luck. And then again in the textbook, the introduction to scientists, carbon dating method is based upon the radioactive decay of naturally occurring C14 and it is developed by Willard Libby. He had been awarded with Nobel Prize in 1960 for this invention. The age of materials determined by this method are published in the journal Radioactive Carbon. Now, fifth one is what? Connecting link. How one phylum connects the other or one class to the others. Three examples given in the textbook: duck-billed platypus, lungfish, peripatus. And students, I added the animation image of these species, how they move around in the next slide. Some plants and animals show some morphological characters by which they can be related to two different groups. Hence, they are called as connecting link, means which helps to connect or know the understanding between one phylum and the other phylum, like in case of reptilia and amphibia or reptilia and aves, right? Now here the example as you are learning is peripatus, characteristic like segmented body, thin cuticle and parapodia-like organs are present, which you can see in this image clearly. And as I told in the next slide, you can see the real species how they move about. Similarly, these animals show tracheal respiration, that is breathing movement, and open circulatory system similar to arthropods, means they don't have closed system, that is blood which is not confined to the capillaries, veins, arteries, they come out of the arteries and veins. This indicates that peripatus is a connecting link between annelida and arthropoda. Similarly, duck-billed platypus, second example which you can see here, lays eggs like reptiles but shows relationship with the mammals too due to the presence of mammary gland and hair. Now as we know, reptiles don't have mammary gland, right, or hair, but in this species it is present. But mammals, they don't lay eggs, they give birth to the young ones, but which does by the reptiles. So it is a link between reptile and the mammals. Third example is what? Lungfish performs respiration with lungs irrespective of being fish. So as we call lungfish, they got lungs. As we have learned, fishes have got gills for breathing, but this particular type of fish has got lungs, even though they are fish living in water using gills, right? So these examples indicate that mammals are evolved from reptiles and amphibians from the fishes, how the evolution has taken place. So now here you can see the animation image of duck-billed platypus, how it moves in water. Here you can see the lungfish, even though it is in water, they have got lungs present, so they can breathe with the help of lungs if they are outside the water body. Then peripatus, you can see the parapodia foot, and you can see they're squirting the some solution from their body region, which may attack the any predator which may come to eat like birds, or which may help in catching the prey. Okay, so see the animation, how beautifully it looks in the animation image. Then lastly, we are learning about embryological evidence. What is embryology? The baby when they are in the fetus of the mother. So you can see all these species when they are in the embryo, maybe first week, second week, they look similar. So we cannot say this is of a bird, or it's of a human, it is of a cat, dog, or fish, right? So see the similarity in the embryo. So we cannot identify in the beginning week of the embryonic stage, but as they grow further, they are then able to be differentiated with their external feature, which we learned the morphology part, right? So comparative study of embryonic development developmental stages of various vertebrates given in the picture shows that all embryos show extreme similarities due to initial stages. As you can see, the first stage, and those similarities decreases as they grow further. You can see the changes taking place. That is what is genes. The genes carry their particular uh, characteristic trait. So as they grow according to the genetic features, they keep on growing and changing, so their morphology changes. So similarities in the initial stages indicate the common origin of all these animals. So all these animals have originated from a common ancestor. Okay, so in the next slide you can see I just placed how the embryonic stages in case of human baby, or as we as a the development takes place in the fetus. So you can see the beginning first few weeks how the baby is present in the human body. Just to add a information for you, the first part the fetus which develops is the heart. So you can see the heartbeat. That's why when mother uh, goes to a clinic for checkup, the doctor checks the heartbeat through sonography. The second stage, how the child is, or the fetus, not the child, fetus is moving inside the mother's womb, and slowly and steadily you can see third trimester, that is first trimester, second trimester, how the child slowly develops inside the mother's womb. So the embryonic stages, this I added for your general information for knowledge, right? Then that ends with the first part of the video, the chapter Hereditary and Evolution. Hope this part helps you to learn and revise the chapter. Okay, so thank you students. So next, the remaining part of the chapter, that is about Darwin's theory, Lamarckism, speciation and human evolution, we'll be learning in the second part of the video.